Raymond Schiffelers from the University of Utrecht, who will talk about this project which is called MINDS, and it's on microvesicles. It's a very interesting, let's say, new field within drug delivery. So we're looking forward.
Okay, thanks a lot. Indeed, this is microvesicle-inspired drug delivery systems. And if you think I'm too old for starting grant, indeed that's true, because in that time the consolidators weren't separated from the starting grants. And if you think you're too old for the consolidator phase, well, I was still eligible, so I cannot help it. Okay, thanks. What it's based on is the fact that plasma, we take our inspiration for these nanomedicines from nature. And there are a number of nanoparticles present in plasma, and they've been identified a long time ago. There are micelles, of course, and there are the well-known lipoproteins, LDL and HDL, in the 10 to 20 nanometer region. And then there is a class of bilayered vesicles within plasma, and they measure from anything between 30 nanometers up to a micron. And they've long been regarded as debris, as parts of cells that have fallen off. But actually, emerging evidence over the last couple of years, last decade approximately, seems to suggest that they have a functional role in transporting molecules from one cell to another, a form of long-distance communication within our body. Since they essentially contain all biomolecules, and this can be microRNAs, for example, enzymes, but also surface molecules, surface receptors, these can be transferred from one cell type to another. And that formed the basis for the idea that perhaps we could mimic nature and modify our liposomes or harvest these vesicles and use them for drug delivery purposes. That's the idea, and this is the concept explained. They are historically known as microvesicles, and that's the term that I adopted, but the field has progressed over the last couple of years, and the term currently adopted is extracellular vesicles. So if you're interested in this subject, look for extracellular vesicles as the term. And they consist of a variety of different vesicles. The microvesicles are actually now regarded as the ones that are shed from the cell membrane, so these ones. And they subsequently travel further. And there are also a class of extracellular vesicles known as exosomes, and they are found in multivesicular bodies, and when these fuse with the cell membrane, the whole cargo, multiple exosomes are released as in one go. They are regarded a little bit smaller, the exosomes. They're about 50 to 100 nanometers, and the microvesicles are more variable, differing somewhere between 100 nanometers and 1 micron. But so far, people have not been able to really separate the two after they are released. So once you are in plasma or in cell culture medium, you essentially always have a mix. And in this case, I use microvesicles as a term to encompass all of these structures. So in principle, any biomolecule can be found and can be transferred from one cell to another. So that was the basis for the idea. So this is the presentation that I gave to the ERC panel, Microvesicle-inspired drug delivery systems. Based on my expertise in delivering biologicals, I made synthetic delivery systems for Si Rna. And what I found out, or when we do rough calculations, I find that approximately 1 in every 10 to the 10th molecules of Si Rna that we inject actually arrives in the target cell. So our synthetic systems are relatively poor. The fact that we do see effects is probably related to the fact that the siRNA is really potent. It's a very potent drug, but the delivery efficiency as such is quite poor. So perhaps or that at least constitutes the problem. They're very potent, at the same time they're fragile, so there's a real need for delivery. And these are the current, for siRNA, we need about 100 molecules per cell to have 50% silencing, which gradually drops off and lasts for about a week. They suffer enzymatic degradation, their circulatory half-life is really poor, and they have a strong negative charge. Well, all of this has been discussed in this conference, of course. And then these papers appear from 2005 onwards, where exosomes could transfer messenger RNAs and microRNAs from one cell to another. And also epidermal growth factor receptor was shown to be transferred from a tumor cell to nearby endothelial cells. So apparently these difficult biological drugs can be transferred from one cell to another and can change also the acceptor cell. So from 2005 these papers are appearing. The current term, or the term is microvesicles, but there are differences. They can range from 30 nanometers up to 1 micron. They contain essentially all biomolecules, and they are produced by activated cells and can change the acceptor cell phenotype. So this offers a new way of communication between two cell types, whereas at least I thought that it was just based on a gradient of biological molecules. There's a donor cell which releases all kinds of mediators that in response to the gradient will more or less activate the acceptor cell. Now we have a package of biomolecules that travels as a quantum piece of information from one cell to another, and it's not diluted, this signal, it arrives intact at the target cell. And that is then the idea for this current proposal. So in principle, as I said, any biomolecule can be there. And when we did, and this is some preliminary work that I presented to this committee, phosphatidyl serine is a phospholipid almost always encountered in these extracellular vesicles and exposed on the membrane, on the outside of the membrane, that's important. And an opsonin or a phosphatidyl serine interacting molecule, lactadherin, which also has an RGD motif in its protein structure. Well, we can see that these vesicles are actually quite strongly taken up by activated endothelial cells. What I try to convince the committee is that we can make these, based on our liposome experience, we can make artificial microvesicles, or we can isolate them, harvest them from cell culture. And what I thought was important to stress is that we should analyze them like we do with our drug delivery systems, analyze their size, their zeta potential, and that this would be important to judge their performance, and then ultimately also study the targeting, of course. Two approaches with two PhD students, one bottom up, the other top down. So starting with harvesting from a bioreactor these vesicles and then try to make them as simple as possible and try to re-engineer these vesicles, or to start with our liposome expertise and combine that into these vesicles. Well, the different phases of the project, obviously going from simpler to more complex and in vivo studies. Something about me. That I was fit for the job. I do not have a perfect CV for that. I don't have high impact factor papers, but well, it didn't stop me at least, and it shouldn't stop you, I guess, because it's something that is important, but not so important that your project could not be funded because of that. And then, of course, to stress something that there were possibilities beyond the direct scope of this proposal, namely that you could, well, first of all, the therapeutic intervention was really what we started here from, but also diagnostic use. Is there a relationship between microvesicle composition and disease, but also the targeting potential to apply microvesicle designs to different molecules and different drug delivery systems. And then, well, this is the place where it all should happen. This is a Department of Pharmaceutics, but there's a lot more infrastructure around this campus. So there was a square kilometer of campus where microvesicles could be a new form of nanomedicines. So that was the talk that I gave to the committee. Currently we're struggling. I wouldn't dare to present our results to a nanomedicine audience because it's really difficult to work with these systems. We are at the early phase of liposome research, I would say. So the target selectivity up to now seems to be limited. The donor cell, acceptor cell combinations, well, so far everything we add to other cells is taken up. There seems to be no selectivity whatsoever. In vivo we see predominantly mononuclear phagocyte uptake, so it travels to the macrophages basically, but there's a small proportion that seems to have a longer circulation half-life, but it's about 5%, and we're not able to really pinpoint what makes these 5% circulate for a longer time. However, what is interesting is that we see efficacious RNA delivery. There's only little RNA inside, but it is really very efficiently transferred. So that's the interesting part. Well, I was quite early on in this wave of interest in these microvesicles. Currently, the NIH Common Fund, with all kinds of other funds, is now allocating $100 million for this phenomenon of transfer of RNAs primarily, based on the fact that they are found in now almost every bodily fluid. So the cerebrospinal fluid, urine, blood, everywhere you can recover these vesicles. And based on some fundamental biological questions like tumor cells are able to secrete large amounts of these vesicles, and they seem to be able to educate or transform nearby cells and make them oncogenic, or at least transform these cells and make them nourish the tumor. And of course, the interesting idea is, would we be able to harness these vesicles, inject them, and let them be taken up in a targeted manner, and by that, well, do what we are all trying to do here and make efficacious nanomedicine formulations. And with that, I think I'd like to end this presentation. My time is up, so I'll take any questions that you might have.
Thank you very much. Questions?
Yes.
Very interesting presentation also. My question is, you are using those microvesicles for drug delivery purpose or Si Rna administration. But there is another way which is probably also interesting, is that in some case, because those microvesicles can play the role to dialogue from cell to cell, from cancer cell to cancer cells, is it also a way that we could investigate to see how molecules, therapeutic molecules or nanomedicine can move from one cell to another and better diffuse into the tumor?
Yeah, it's actually, there are a couple of studies that suggest that certain molecules are able to hitchhike along this pathway and actually gain access to the tumor by hitchhiking along these vesicular pathways. So try to gain entrance into these multivesicular bodies and subsequently be excreted in the nanomedicine formulation. Indeed, that's one of the things that people propose. Another one is to try, and since it is a therapeutic target in itself, microvesicle production, it appears to be involved in cancer progression, so why not attack the vesicle production? That seems to work. And what we're currently doing is in vivo experiments. We have, what we're trying to do is look at the delivery efficiency and communication between different tissues. We have tumors implanted with Cre recombinase and a lock site in front of a confetti fluorescent protein on the other side. And actually we do see that the other tumor becomes fluorescent, and the only way why that is possible is by recombinase arriving there. We cannot detect the recombinase protein inside vesicles, but we can detect the messenger RNA. So, well, that's currently work in progress.
More questions?
No.
No? If not, I think we'll go for lunch. Thanks for staying out so long. Sorry for taking part of your lunchtime. Let's thank the last speaker again.